Good morning, everybody. It's a real pleasure to be here and an honor to be welcomed into the FOXG1 family and community. Sue is a very tough act to follow because the science is so exciting, really incredible. And honestly, without that science, without the work from Sue, Jay, the UB team, probably the work that what you'll hear from me, what you'll hear later from Ellie and Kopika would be almost irrelevant, right? Because it's all based on the scientific foundation. So what I'm gonna try to do in the next 20 minutes or so is give you a line of sight or an orientation to what it takes to take the exciting science that you just heard about and move it forward through the process that will take us into and through clinical trials, and where we are right now along that journey. So this is, what I'm trying in this slide is just schematically give you a sense of this is the research bubble, okay? So usually in academic research, and Sue knows that more than anybody else in the room, including myself. Here, usually there's a certain cycle that happens in academic research where there is a certain scientific question, curiosity, could be disease related. And there's a research conducted around that, trying to understand the biology, the mechanisms, what's happening in the body, in the cells. And then try, based on that, to identify a potential target that we can go after with a therapy. And we come up with a hypothesis. If we modulate this, then we can help with that. And then we build models, everything that Sue described. We know what the problem is. There's the interrogation of the mechanisms. And then we build models, mouse models, cell lines. And then we test the hypothesis. We take these models and we come up with our therapeutic theory and we test it. And this is what we call here POC, which stands for proof of concept. This is where we test it in a model, and we see whether we move the needle or not. Usually, in the academic cycle, this is where it ends. This is success. We have proof of concept. We move the needle in a model. We publish. We present in conferences. We move on to the next project. This is sort of a pinnacle in the academic space, but this is not yet a drug. Now, this is clinical practice. Okay? We don't feel well. We have symptoms. We go to the doctor. We get a diagnosis. Based on the diagnosis, we might be treated. Depending on the treatments that are available, we may be prescribed an approved treatment. It may be a treatment to manage symptoms. Maybe it's a disease-modifying treatment that really goes after the root cause of the disease. And then if needed, there's chronic treatment and management of the condition. And maybe that becomes standard of care. This cycle, which whenever we go to the doctor, that's what we go through, is based on approved drugs. This is a cell line and a mouse and a proof of concept. But how do we get from here to here? So I think the answer is most of us know it. It's pretty straightforward, or it seems to be straightforward. It's called clinical trials. Okay. So let's talk about clinical trials. We already talked about research. So in clinical trials, there's a different cycle. We need to address certain questions. Who's going to be our patient population for the trial? Doses. How do we dose? Where do we dose? How much do we dose? How frequently do we dose? What do we test for? How do we measure success and treatment effect? We measure treatment effect, we measure safety, we measure other parameters that are needed to know how the drug behaves, and then we get data, we get results. And then if it's good enough, we may go to FDA or another agency and say, hey, this is good enough, we want you to approve it. And they may or may not approve it. And if yes, great. And then we have an approved drug. And if not, then we go back to the cycle and we redo maybe a clinical trial until we figure it out. So there's a catch, right? So we need clinical trials, but in a clinical trial, we actually need a drug. This, which is usually where the academic research kind of ends, this is not a drug yet. So clinical trials bridge between research and the clinic, but we can't jump from here to clinical development because we don't have a drug yet. So we need another step along the way, okay? And that step is the translational science step, right? That's where we take this and we actually try to bring it to the point where it is a drug that can be tested in humans. This is where, there's a fence here, this is where a lot of programs die. And part of the reason that they may die is that there is a certain tendency to, what I call here, toss the molecule over the fence. And what that really means is you have academic experts, and they are really good at this. And then you have drug developers, and they're really good at this. But they don't really know each other's language because they speak different languages. They're both smart, they're both capable, they know what they're doing, but if you stop here and say, okay, we're done, we'll publish our paper, you guys take it from here, you're putting the program at risk. What we really want to do, and we're doing it in real time, is establish a continuum where we're not tossing a molecule over the fence, where in the research space, which Sue and the team are leading, there is clinical input, clinical considerations that are already integrated into the research plan. And as we are transitioning into clinical development, research continues to be a foundation to what we're doing and how we're doing it. So there's a dialogue. I wear the development hat. Sue wears the research hat, right? But those are just hats. What is really going on here is a continuum, a dialogue, a collaboration, a partnership that makes that process, that transition in the translational space, I think, a lot more efficient and really sets us up for a better probability of success down the road. There are a lot of steps that need to happen before we can reach this to reaching the clinical trial itself. So here I'm just giving you a snippet of the stuff that we're doing right now. And we are in this space of translational science. So where are we? Proof of concept, you just saw, Sue. Fantastic proof of concept in models. Now we need to get it into the clinic. This is the stuff that is happening, ongoing, will continue to go now and through 2025 to bring us to the point where we can go to FDA and other regulatory agencies and say, we're ready to go. Do you agree? And the hope is that they will say, yes, move forward. Green light, tested in humans, right? So this is just really the tip of what we need to go through. So I put it in different buckets here. There's a lot of non-clinical work. This really focuses predominantly on safety. We want to make sure that as we enter the clinic, we have a good sense of safety. Right? Safety first. So we need to understand safety. We need to understand dosing. A lot of this work is happening here. Manufacturing. We need to make a drug. We need to partner. We're not manufacturers, right? We need to. Anasha mentioned Charles River. We're partnered with them. A lot of process to make a drug that can be tested in the clinic. That's already ongoing. Bioanalytical, I'll try to not use curses, but really what this means is establishing tools that help us make sure that the quality and accuracy of our drug properties can be measured and verified. On the clinical side, some of you are part of the registry. Some of you have been participated in the natural history study. Some of you participated in what's DCM here, disease concept model, what Eli and Kopika will talk about a bit later. And then a whole lot of other activities, outcome measures, biomarkers, other curses that I'm not going to utter here. But really, all this is a lot of work that needs to happen between now and clinical trials in order to be ready for these clinical trials. Later on, together with Nasha and with Eli, I'm kind of already highlighting here this word here, outcomes. We'll talk about that later, because this is where heading into 2025, we're actually going to need your help. And then there's the regulatory side. Ultimately, to move into a clinical trial, we need to talk to the regulators. FDA in the US, other regulatory agencies in other countries. We already had a very successful first meeting with FDA. This is an Interact meeting which we had in July that went exceptionally well, and we're gearing up toward our next interactions with FDA. Ultimately, all this work will need to will culminate in us being able to submit what we call an investigational new drug application, or IND. This is in the US. Outside of the US, it's called CTA, which is basically a clinical trial authorization, that we can move forward with a clinical trial. So all these are discrete efforts, and they're groups of people and teams and partners that are doing each and every single one of those. So all this, I won't do it to you, but each of this we can blow up and give a whole slide of explaining what that means. But while these are discrete efforts, they're also interconnected. So there are dependencies here. One effort relies on another, and a third, and a fourth, and a fifth. So if you draw the connections and relationships between them, then you get this sort of spaghetti bowl, right? And this is just to show you that it's a little complex, the process. And if you compare this to an orchestra, then if it's not coordinated, you may have fantastic players with top-of-the-line instruments. If not playing together, you have a cacophony, right? It's going to be a disaster. If you coordinate them and they play together in sync, feeding off of each other at the right time, in the right place, you have a beautiful musical, piece, and you have a symphony. And our goal is to make sure that this is a symphony, right? And so all this work, all this coordination, all these moving parts are happening right now. How do we do it? We do it, because we have a great team. We have strategic partners, and we have a shared goal. At the end of the day, we have a shared vision, and I think that this is what drives everything. And in terms of our partners, first and foremost, it's you, right? And you're partnering with us in so many different ways, and we're going to need your partnership also moving forward ahead toward clinical trials. The Lee Lab, of course, Charles River, consultants that we're working with. We have a great scientific, I see that the slides got a little, squished here, but we have a scientific advisory board. The clinicians, some of them are in the room, amazing clinicians that we're working with. And we have our sort of legal support that we're working with, and of course the donors, which we talked about and you'll hear more about. So maybe in summary, in order to move from research to clinical practice of an approved drug, we're going to need to kind of make some room there and conduct clinical trials. But conducting clinical trials is also not trivial. We need to get some work done before we can do that. And this is where we need the translational science, which is where we are today. Okay? And at the end of the day, what we really want to achieve, our ultimate goal, would be to take this work of a preclinical proof of concept that Sue described, and at the end of that arch, have a disease-modifying treatment that becomes standard of care for FOXG1 syndrome. Thank you.